And our next speaker is Dr. Teo Wee Fei, or Aaron, as we know. He's actually a Malaysian, but he graduated with his PhD from Kasetsart University in Thailand in 2020. And he's also a microbiologist. His PhD is also in microbiology. So he's previously worked as a temporary teaching assistant for Division of Microbiology in UM. And I think Dr. Annie, he's worked with Dr. Annie, right? And I think she's here in the audience today. Before he went to do his PhD in Thailand. And we're very happy to have him back in UM as a postdoctoral fellow now in Sabah. His research interests include bacterial taxonomy and classification, as well as bioprospecting actinobacterial resources. And he's actually contributed a book chapter to Bergey's Manual of Systematics of Archaea and Bacteria. Now, any student of microbiology, I was an undergraduate in microbiology as well, knows Bergey's manual very well. So that's also, of course, has several research papers as well. He also peer reviews research papers for Annals of Applied Biology and International Journal of Systemic and Evolutionary Biology. So now I'll invite him to present his paper, and he's chosen the title Kantulee Peat Swamp Forest Associated Actinomycetes and their Plant Growth Promoting Potential. Take it away, Dr. Aaron.
All right. Thank you very much, Prof. Jenny, for the kind introduction. Let me share the presentation. Right. Very good morning. I'm Aaron, and today I'll be sharing with you my PhD research experience conducted in Kasetsart University. The title of my PhD is Kantulee Peat Swamp Forest Associated Actinomycetes and their Plant Growth Promoting Potentials. So I will be sharing some information on the peat swamp forest and the motivation why I'm looking at actinomycetes as plant growth promoters. I would also briefly discuss the strategies involved in the isolation of actinomycetes. Then I would share some of the diversity of these isolated actinomycetes and their identification by 16S rRNA gene sequence analysis. Next, I would characterize these potential plant growth promoting actinomycetes via in vitro and in planta assays. And last but not least, I would also share some information about describing new species isolated in during this study. So peat swamp forests, generally the soil from peat swamp forest is waterlogged with a mixture of wood with different degree of decomposition. This in turn would develop three distinct microbial communities within the peat swamp, namely the first aerobic active microbial communities on the peat swamp surface that decompose the organic litters. Then they are the anaerobic microbial communities deeper in the peat column. And lastly, they are the dormant communities that became dormant when the condition is not favorable, and when you drain or aerate the peat, then they will return to be active again. So what could we learn from the microbial forest, natural forest system that could help in the agricultural system? In the natural system, as shown in the figure on the left, consists of a diverse species of plants that could in turn produce a variety of root exudates and plant litters. The nutrient absorbed from the soil would eventually return back to the soil system. Right here. This closed circuit of nutrient cycling would support a variety of microbial communities that both the good and the bad at a balance. This complex interaction between the plant and the microbial community become less sensitive to sudden changes like disease infestation or sudden environmental change. In contrast, the agricultural system, as shown in the figure on the right, consists normally of monoculture and very often clonal plants that in turn would produce limited variety of root exudates. and plant litters. And furthermore, the problem is compounded by human intervention with fertilizers and pesticides that would eventually select only a typical group of microbial communities would proliferate. This in turn will result in a low microbial diversity, thus simplifying the interactions between the plant and the microbial communities. These simplified microbial interactions with the plant become vulnerable to sudden changes like disease infestation and sudden environmental change. So the idea here is to provide a plant probiotics to promote the general well-being of plants in the agricultural system. Why the need for these plant probiotics? Firstly, we know that the complex interaction between the plants and the microbial communities could buffer the biotic and abiotic stress. However, not all farms and plantation allows you to change the crops every year. So it would make sense then only to add the microbial to enrich the microbial community. Secondly, there are the problem of abiotic and biotic stress. Of course, you could modify or genetically modify the plants through breeding or genetic modification. Alternatively, you could also inoculate the system with disease suppressing or stress elevating microorganism to reduce the stress level. And lastly, of course, we have the resource use efficiency. Plants or multiple plants could be used to increase the efficiency of resource use, or alternatively, the plant probiotics could also increase the nutrient retention, cycling, and acquisition of nutrient for the plants itself. Now that we know why how plant probiotics could help, I would like to pitch for why Actinomycetes is selected as my research of interest. Firstly, research has shown that the Actinomycetes could react to plant stress signal and produce secondary metabolites to alleviate the stress. And secondly, Actinomycetes could also act trialectically by modulating the microbial community by means of antibiotics or siderophore to suppress other unfriendly microbes. Another thing is about Actinomycetes is relatively the genome is large, and they dedicated about 10% of the genome to production of secondary metabolites, which means they on their own could produce a wide variety of secondary metabolites that could be exploited. And furthermore, the Actinomycetes are spore formers, meaning that in the downstream application, the formulation for Actinomycetes as plant probiotics would be much simpler. From production to logistic to storage, Actinomycetes spore can be formulated as a dry, stable, powdered products. The characteristic of Actinomycetes as spore formers is the very reason that the isolation of Actinomycetes is different from other bacteria, and this character can be further exploited. Before I begin, let me briefly mention that the peat soil samples were collected from the southern region of Thailand. Samples were collected from the primary as well as the secondary sections of the forest reserve. As typically for the isolation of bacteria, soil samples are serially diluted and then plated on selective agar. However, the isolation of Actinomycetes, typically the soil samples are air dried to reduce all motile microbes. And during the serial dilution, the suspension were pre-treated at 55 degrees Celsius for 10 minutes. So by now you should notice that one could employ these isolation strategies in formulating plant probiotics. The figure showed the Actinomycetes on the isolation plates. Blue are Actinomycetes and the red are the fungi. Yes, Seba staff had raised an hand. I'll continue that. So a total of 75 actinomycetes were selected. Of these 75 actinomycetes were then subjected to phenotypic characterization. The actinomycetes were grown on ISP2 and ISP3 media to observe the color, diamino acid cell wall composition isotypes were broadly used to separate actinomycetes into streptomycetes, the commonly isolated streptomyces, or the less commonly isolated non-streptomycetes. And lastly, the pH tolerance for growth. So the results show that there are 57 isolates of streptomycetes. 50 of it are acidotolerant neutrophiles. Non-streptomycetes are 18, 14 of which are acidotolerant neutrophils. Streptomycetes could be further grouped into 7 color groups. So they have a wide array of color, red, green, yellow, and while the non-streptomycetes can be further grouped into 4 color groups. Next, the selected representative isolates were subjected to identification to the nearest taxa based on 16S gene sequence analysis. Results show that these representative isolates were members of the genera Amycolatopsis, Nocardia, and Streptomyces. So moving on to the next section, assessing the plant growth promoting potential of actinomycetes via in vitro and in planta assay. First, the first assay is the siderophore production. Siderophore production was tested on pH 5 and pH 7. Shown in the figure is the same isolate.
Sarina, please turn off your mic. Thank you.
Shown in the figure is the same isolates on 2 different pH, displaying different degree of production. So as you can see here, there's no production, there is production, and likewise at pH 7 for the same strain, there's a higher production. So as shown in the bar chart, there are more isolates with activities when tested at pH 5 than in pH 7, but the production index at pH 7 is much higher. Second assay is the phosphate solubilization. Shown in the figure is the isolate displaying different degree of phosphate solubilization. So there's no activity here and there's activity here. As shown in the bar chart, there are most isolate display low or no activities. So most of them don't solubilize. Some dedicated actinomycetes do solubilize the phosphate. And lastly, the isolates were assayed for their antagonistic activities against different fungi. Shown in the figure, the inhibitory effects against the radial growth. So the presence of actinomycetes inhibit the radial growth of the fungi against the place where the actinomyces is. So by collating all the in vitro results, 1 Amycolatopsis, 2 Nocardia, and 14 Streptomyces strains were selected based on their various activities, the siderophore production, phosphate solubilization, and antagonistic activity against rice fungal pathogens were selected for the next test. So for the next test, the isolates were grown on ISP2 broth and shaken. Cells were harvested. At the same time, the Thai jasmine rice seed were surface sterilized. The cell and the seed were then soaked together for 18 hours to bioprime the seed, and then the seed were germinated on a petri dish. And after 3 days, we evaluate how many of the seeds were germinated, and if they germinate, what is the root length, shoot length, and the percentage of germination. 4 strains, namely the Amycolatopsis acididurans, the Nocardia, and 2 Streptomyces strains were selected based on the percentage of germination, shoot length, root length, and the vigour index for the next test, which is the pot experiment. For the pot experiments, similarly, the cells were grown in broth form, cells were harvested, seeds were surface sterilized, cell and seed soaked, bioprime, 3 days germination, pre-germination, seeds that germinate with similar root and shoot length were then selected for the pot experiment. After 14 days, the percentage of seed being able to establish in the soil and the seedling length, which is the shoot length and the root length, were measured. The results show that the effect of Actinomycetes bioprime on rice seed germination show that the untreated seed have the vigour index of around 2000. Nocardia don't perform very well. In fact, most of the seedling wilted after the 7th day. The Streptomyces perform quite okayly. The most promising strain is the Amycolatopsis Acidotolerant K13G38. The last part of my presentation would be description of the novel species. These would probably overlap with what being said by Dr. Kantulee. So generally, a strain with 16S RNA gene similarity below 98.7, then you have a good chance of a new taxa. So K13G38, K81G1, isolated from this study, had the higher similarity of only below 98.7. Then building the phylogenetic tree show that they are very distinct. So to describe a new species, you need two main data to be collected, which is the genotypic data as well as the phenotypic data. So for the genotypic data, the Amycolatopsis, generally we have to, the genus Amycolatopsis, the genome size is ranging from 7 to 11 mega base pair. GC content is about 68 to 72. The ANI threshold for a new species should be below ANI of 97 or DDDH below 70. So evaluation of the strain isolated from this study and the closely related species with validly published name show that they are all within the expected range of the Amycolatopsis. And the pairwise analysis of genome similarity show that they are all below the threshold among each other. Phylogenomic tree also show that both strains are closely related. This is expected as both strains are isolated from a very similar environment. Phenotypic description is part of what you have to do for a species description. So expectedly, Amycolatopsis mature cells would grow into rod-shaped fragment, and this is demonstrated in the scanning electron micrograph, showing that the filaments of Amycolatopsis will break into small short fragments. So as the result for the carbon utilization and substrate degradation, usually used to show the differences between the species. These closely related species have different optimum pH for growth, therefore were tested at different pH, pH 5 and pH 7. Expectedly, the result also show that the carbon utilization and substrate degradation profiles may change with pH. The last but not least, as part of the phenotypic characterization is the chemotaxonomic, which is the use of chemical biomarkers for Amycolatopsis. It is expected to have mesodiamylic acid, aminopimelic acid. They have arabinose and galactose in their whole cell sugar. The presence of PE as a phospholipid, and the minaquinone is either MK9H2, H4 or H6. So as shown in the result, in our experiment, the two strains fulfill the profile expected of Amycolatopsis, and as well as the closely related strains. The fatty acid also show similar profile. So for the description of new species, you need genotypic and phenotypic data. So the genotypic and the phenotypic data of both strains is consistent for that of Amycolatopsis. That's why they are placed under they are classified as Amycolatopsis, but they can be distinguished from closely related species. K13G38 is then described and proposed as the new species with the new species name Amycolatopsis acididurans. It's published in Journal of Antibiotics. It's currently waiting for validation in IJSEM. The expected validation is the coming September 2021. K81G1 is proposed as Amycolatopsis acidicola and been published in IJSEM. Once you publish in IJSEM, you automatically be validly published. So in summary, Actinomycetes have been isolated from peat swamp with plant growth promoting potentials. 25 isolated Actinomycetes that can be grouped into 11 groups. Identification by 16S rRNA genes show that there are two represented by Amycolatopsis, Nocardia, and Streptomyces. The plant growth promoting Actinomycetes 17 was selected for rice biopriming, and the pot experiment showed that Amycolatopsis acididurans K13G38 is shown as the most promising strain. The new species, both of these strains isolated from this study, were proposed as new species, one of which had been validly published. So from the PhD study or the PhD work, the general information had been published in Chiang Mai Journal of Science. The new species is published in these respective journals. So while I'm in Kaseh Sat working with my supervisor, I had the fortunate luck to have to co-author and contribute in Burgess Manual of Systematic Archaea and Bacteria for the chapter for Alocarcinaria, as well as helping my junior on publishing her new species. So with that, I would like to acknowledge my supervisor, Dr. Kanika Dongman and Dr. Nantana Sisuk, who had helped me in my PhD work. The work was supported by the Higher Education Commission, and my PhD was supported with the scholarship from the graduate schools. With that, thank you very much.
Thank you very much, Dr. Aaron. Another very nice and on-time presentation. So we open for questions. And like before, if you prefer to type, you can type in the box, or if you would like to ask your question out loud, you can raise your hand and we'll invite you. I also need to remind you to fill in the survey form. And also, you know, these have been great seminars that have been recorded, and we'll be putting them up in our Facebook page, sorry, a YouTube channel, right? I think. So if any of your friends or colleagues have missed the seminars, they will be published online shortly. There was a comment just now. Is it Pitri want to ask some questions?
Right. I think Dr. Fitri's question would be, I have a question which are how does this Actinomycetes benefit the plant environment or even us, and why are their growth being stimulated? Is it to produce more Actinomycetes colony? I think that would be more appropriately for this. Right, so in general, Actinomycetes can grow if you inoculate in the environment itself. There exists Actinomycetes, but their population is normally not the predominant taxa in the soil system. So by inoculating or adding more population at a proper interval, you could enrich this type of taxa within the soil system. It works very much. Probably that's why I use the word probiotics, plant probiotics. It's like how our gut works. You drink Yakult or Vitagens or probiotics, it will generally promote the good well-being. And this is the very same thing. That's how it works for the soil system as well. You could add in more actinomycetes. Some of the actinomycetes could continue to grow, but you can always push the threshold of the population up to keep it at the optimum population so that it could benefit the plants. How it benefit the plants, they interact with the plant mostly. They do produce plant phytohormones, and the phytohormones can be used by the plant itself to reduce the stress. They regulate other microbial communities, meaning that they suppress every other taxa. If the taxa of the pathogenic taxa is present, actinomycetes normally by default they would have suppressed their presence as well. They don't kill them, they don't eliminate, they don't cause the extinction of the pathogenic normally. They would suppress them to a very low number. With this suppression, normally the disease will not be manifested. So I think that's one way that the plant growth promoting actinomycetes could help.
While we wait for more questions, I have one. You surprisingly showed, or surprising to me, the nostoc it actually grew worse, right? The rice grew worse.
Yeah. Yes.
Is it a nitrogen fixer?
No, it's not a nostoc. It's a Nocardia. It's a different.
Okay, that's where I'm confused. Okay, so any of these bacteria nitrogen fixing?
It was not tested. At least it's not tested. And these are not known to be at least this group that I'm working with is not known. There is other group of actinomycetes are known to fix nitrogen. The Frankia, if I'm not mistaken, the Frankia. Yeah. Oh yeah, I wanted to add to what Dr. Nazrin asked previously on, can you describe new species on the basis of genome and 16S? Dr. Kantulee had added that on this genome data is not enough. So you have to put in the phenotypic data, which is correct. But what I would like to add is that even if you do collect the data for the genotypic data as well as the phenotypic data, you would still need to have the name validly published. Because for bacteria or for prokaryotes, it has a central registry system whereby if you propose a name, the name needs to go into that registry so that the name would be valid or correct. So that's another issue where a lot of people describe the new species, publish it outside IJSEM, and then they don't take another step to have the name registered or indexed. Then the name is not validly published. So you can propose, but the name will never go into the proper system. So yeah.
I have another question.
Thank you, Dr. Aaron.
Okay.
Is that name validly published, Kantulee?
Yeah, I think last 2 months or last 3 months, I forgot. But if let's say you check on the LPS and the LPSN, it is already very published.
Yeah, congrats.
Yeah, that is an important point, and I think maybe we have Dr. Zaktil in the audience. We also, when you look at the open science, the validation of sharing and having your information. So sometimes it's good to look into that. Sometimes publications, and especially if we are able to provide open access, this is a really important thing for your science to be used by other people and hopefully have some benefit in the world beyond just making a thesis or a paper, which we all hope, right? I think another thing that is very difficult here is that these are all presumably working in communities as well. Like you mentioned, even reducing the diversity among the plants is going to reduce the diversity among the microorganisms. So this can be one of those things. Are there people who study on actually using, because people do intercrop with legumes and other plants to try to improve the microbial and the soil conditions. Is that possible with something like rice, or is it only possible with bigger crops like trees?
That's the right question. So not all the time that the suggested way of improving the not, it doesn't always work in all farm system. So intercrops, yeah, but like Prof had rightly pointed out, it doesn't work in the rice system. Changing the plant every now and then doesn't work in palm oil or durian because they need years to grow, and then you only can harvest. Can't change it every season of harvest. So in a case where the plant cannot be changed, then the next best thing possibly is to inoculate with plant probiotics.
Okay, do we have any more questions? We actually have run a little bit over time, but I wanted to give Dr. Aaron a chance to take some questions. Okay, there are two questions here from Rossini. Can you see those?
Yes.
For anyone else, please refer to the chat box. You can see the questions.
Actinomycetes were mentioned to exhibit antifungal and antibacterial properties. Does this microbe have potential for medicinal or animal treatment? The direct use of Actinomycetes itself, no. But their product, yes. Actinomycetes are prolific as antibiotic producer. Some of the antibiotics circulating in the current consumption of humans is derived from Actinomycetes. In the identification of novel strain, what are the key details you should look at? The key details would, you need to basically, you should start with this. You would want to start with this. Is the 16S below 98.7% similarity? If there is, then you would like to look at these two types. You would like to collect these two data. So once experience shown from myself and Dr. Kantulee, you have to collect these two data, present it, and hopefully it's sufficient. The reviewer is satisfied with the description, then you get it validly published. The name will stay in the system for as long as forever.
Okay, so I think we're going to wrap it up now because we are actually over time. I would like to remind everyone, please, to fill in the survey form. Perhaps that can be put back in the chat box again by the admin. And thank you all for your attention, for being here, for the very stimulating questions, and most of all, thank you to our two speakers. They are just still new with us, and we are expecting them to do great things together with us and all of you in Sabah. And I wish everybody a good day and enjoy the rest of your week, and stay safe, everybody.